All right. Well, good morning. Thank you for coming early. I'd like to start off by thanking the organizers for what's been a great meeting, and thank you for the invitation. My talk today is it's called Mining Differential Correlation. It's a bit different in spirit than some of the talks we've heard. That it's going to be. I'm going to present a few summaries of some other work that we're doing at the end of the talk, but this talk will be more or less theorem-free, so I hope that's maybe good for the morning. Maybe it's better for the late afternoon, but so be it. So a brief outline. I'll talk about what the problem is, and I'll illustrate it with a couple of examples. And essentially what we're looking at is we have data collected under two conditions, and we want to find a set of variables that is correlated or relatively highly correlated under one set of conditions, one condition, and not so correlated under the other condition. That's basically the problem. We don't know in advance what variable set or sets we might be looking for. So our approach to this, which follows on some work we've done before, is to take a testing approach. And for that we need p-values, and the only sort of theoretical result I'm going to have here is the central limit theorem, which is really not much more than an exercise. And then the key idea is going to be the iterative testing search procedure, which I'll speak about at some length. Then I want to talk a little bit about competing methods. This is a question that's been looked at in various ways in the literature. We're not the first people to look at it or to try to tackle it. I'll give a couple applications, and then at the end here I have bonus material, and that's just referring. I'd like to spend maybe 10 minutes talking about some other work that's gone on in, I guess, our group, if there's such a thing. So I've been collaborating for a while with Shankar Bhamidi, who's a junior colleague of mine, a great probabilist, and we've been working on a number of these things together. The work I'll describe today is joint with Kai Zhang and Kelly Bodwin, and the work at the end here, I'll give references for that later. So please interrupt me if you have questions. I'm hoping that I will not be rushed, and I'm happy to entertain questions if you have them. So if something's not clear, just let me know. So here's the problem. In fact, maybe I should even, sorry. That's the problem. So I'll maybe it's better to show the picture first. Basically, I have, these could be, I've got my samples organized along the top here, and I've got variables, which could be genes. So we can think of this as perhaps a bunch of gene expression measurements of patients who are sick, a bunch of gene expression measurements on patients who are healthy, and those are our conditions. We're trying to find a set of variables A that are correlated, highly correlated under one condition and less correlated under the other. So the criterion is very simple, and the problem, at least in principle, is very simple. The difficulty comes in the fact that we don't have a pre-specified set of variables A. So we have to figure out what A is, and A doesn't have to be an ordered set, is the way I've drawn it there. It's any subset of my variables. So that's basically what the question is. All right, so in a little more detail, we will assume that I have the number of variables is constant. So in each condition, if the gene expression measurements, I'm using the same technology, so I have the same number of genes that are measured. I'll call that M. And I have two experimental conditions. One could probably generalize this to more, but I think it would be, well, it could be done. I'm not sure if it would be very elegant, but it could be done. So we imagine we have N1 samples under the first condition. Each sample is some random vector, it's m-dimensional, and it has correlation R1. And we're not really concerned about the mean behavior here, so the correlation is what we care about. Under condition 2, I have N2 samples. Each of those, again, is a random vector and Rm, and the correlation of those vectors is R2. So to say what we're interested in, which is not entirely obvious, let's look at this thing I call delta IA. So this is the aggregate difference of correlations for if I take a single variable I and I consider, on the one hand, a single variable, and on the other hand, a set of variables A. So it's just the sum, and what I've looked at is the difference of the two correlation matrices, and I'm simply summing that, and I is fixed, and I'm summing over all the variables J and A. Okay, so that gives me an aggregate difference of correlations for variable I with respect to the variables in A.
Can't vary the N1 and N2.
Well, they're given to me.
So N1 could not be 1, for instance?
No. I mean, in our no. So what we're assuming here is that we have moderate. If our sample sizes are too small, I don't think any method is going to work particularly well. And we're not given a network, by the way. We're just given a vector. So for us, n1 and n2 should be, 15, 20, or greater. That's usually enough, depending on the setting. So our goal here is to identify what we sometimes call a differentially correlated clique. So it's a kind of self-consistently differentially correlated set. So here's the idea. So I have a set of variables that has the following property. Every variable in the set is differentially correlated in the sense that delta IA is greater than 0, right? And for every variable outside the set, this quantity is either 0 or negative. Okay, so that's my definition for what I'm after. So, and we'll see when we talk about the algorithm for this that we're actually going to be looking at a fixed point. So there's a kind of fixed point character to this, right? So that it is or what you might call stability or self-consistency. Everything in A is nice. Nothing outside A is nice with respect to A. Okay? So in that sense, A is self-consistent. All right, and then we want a certain sort of minimality. If we don't want to have a set A, which is really a group of differentially correlated sets, we like to find A's that are as small as possible. This ensures that they won't be too small. Okay? And we'll see that in methodologically speaking, we address this, and how we address this, I think, is a little bit awkward, but it works. So
Sorry, I
yeah.
still don't. What's the I?
It's a variable.
A variable.
Yeah, it can be in A or not in A.
And it's a variable, an m-dimensional variable.
Nope, it's going to be an n1 plus n2 dimensional variable. I mean, it's a variable, so I is going to be, let me go to the picture. Thanks for asking. So I is going to be, I corresponds to a row of this matrix. Okay, so I'm looking for correlations across my samples. I'm not interested in the correlations between samples, but I'm interested in correlations of variables. So I'm interested in the correlations of my rows. So A will be a set of variables, as indicated there.
I mean, the correlation is something between something, and correlation with what? Or
Yeah, so here, what I'm looking at essentially is the sum of the correlation of, you can write this as R1 ij minus R2 of ij. I just try to write it a little more compactly, right? So I'm looking at the correlation of the variable I with variable j, and looking at the difference of those correlations across.
It's just difference
Is that
of the
okay?
I with
correlation.
J, yes.
Difference of the covariance matrices, right?
I with
Yeah, is that okay?
But
Thank you for asking.
These are numbers.
Yep, everything here is,
Are real numbers.
yeah, this is, there's no Banach space value to anything here. So this is all, right, it's 9:30, we're keeping it, so, yeah, this is really, this is super simple. There's nothing complicated about it. Is that okay? So again, just to I hope the illustration is helpful. So the variables in A here are, generally speaking, if I look at their pairwise correlations, add them up, right, relatively high. In the other condition, look at the pairwise correlations, add them up, not so high.
And these are covariances or correlations?
Correlations.
So you divide by the variance.
Yes. Okay, and we don't want, I don't think this would be as meaningful if we were considering covariances. So
No,
there seems to be
just
good reason.
making sure.
Yes. Is that, everyone okay?
And the R1 and R2, are they given to you, or do you estimate that from your data?
We're going to be, ultimately we're going to estimate them, and we make no assumptions about them, which is going to be important. We don't want to assume that they're sparse. We don't want to assume that they're banded. So again, we're probably asking, we're asking perhaps a less sophisticated question, so as a result we can make sort of less sophisticated assumptions. Or I'd like to say, maybe more realistic assumptions, right? But right, in the end, I'm not going to show you a fancy theorem with rates of convergence or anything like that. So it's a trade-off. Any other? Okay. So so our goal, of course, we want to do this with data, right? We don't have the true correlation matrices, so we're going to do the obvious thing and look at the empirical correlation matrices. And just to reiterate a point I made before, the variable sets are not specified in advance. So any subset of variables of any cardinality, regardless of how they're ordered, is a candidate. Okay? So obviously an exhaustive search is not something you can do when M is large. All right, but you've heard that before. Okay, so we have to do something special, I guess. Now, and here I will apologize again, at least for the first 19 slides. This is the only time the word network will appear. All right? So one could, of course, look at the empirical correlation matrices, threshold them, get a network, and think of this in terms of networks, but that would be wasteful, right? Because when you threshold, in that case, thresholding the correlation matrices is throwing out a lot of information that would be useful, right? So if you like to think about it, you can think of this as it's sort of, I didn't quite know what to call it, differential analysis for weighted networks, but it's local, right? So if you're given two weighted networks, my question is not, are these the same, but is there some community or subset of those networks, right, where the two networks differ? That's essentially the problem that I'm asking in a very special case, right? So no claims to generality here, but if you want to think about this more generally, that's what we're doing. And this could be a really great thing to study in more generality, but we haven't done it. Okay? So non-assumptions. As I said, the virtue of asking simple questions and giving relatively simple answers is that we don't need to make, and we don't want to make assumptions about R1 and R2. Because we found that in practice, if assumptions are often violated, right? Even ones that seem pretty reasonable. So we're not assuming that R1 and R2 are diagonal, banded, or sparse. We're not assuming that the difference of R1 and R2 is sparse, diagonal, or banded, right? So they are what they are, okay? Really important, I mean, differential expression, right, which is, if I can go back to my picture again, right? So this has been considered, this is kind of what drove the development of false discovery, right? Early in the days of gene expression, people said, you know, I had gene expression measurements here and here, and I wanted to find variables where I saw a significant difference in expression from one condition to the other, right? So people did a ton of t-tests, or maybe they did tweaked t-tests, and then they wanted to figure out, you know, which of these genes were significantly differentially expressed. That is what I would call a first-order analysis, because you're only looking at individual genes one at a time, right? What we're doing here, and what networks are about at some level, is second-order analysis, right? We're looking at relationships between pairs of objects, right? So this is fundamentally different from differential expression, a point that I will make later several times. So you may be tired of hearing about it by the end of the talk. All right, but that's important. So we're not doing that, and we don't want to. Let's see. And so we've tried some applications. We have some collaborations with people in genomics at UNC, and this has helped them, or at least they seem happy. And of course, when you have collaborators, you have to judge their happiness. So there's like the average happiness when you bring something to them that seems sensible, and then there's that sort of extra level of happiness where their eyebrows go up slightly, right? It depends on your collaborators, but we've reached the eyebrow going up level of extra happiness with our collaborators, so that makes us feel good. I tend to be critical, so I'm just trying to ask them, are you sure this is interesting? And they say, yes, it's interesting. Are you sure it's interesting? Yeah, it's interesting. So I think some analysis I'll show later might actually make its way into a paper, so I guess that crosses some threshold. All right, and I'm just sort of blowing smoke, but it seems like this might be useful in economics, but I don't deal with economic time series, so I better not say anything more about that. So here's, just to give you a view, now here we're not looking at the data matrices, but we're looking at the correlation matrices. So this is a typical thing one sees. We looked at a subset of some gene expression data. These are two cancer subtypes, HER2 and luminal B, the breast cancer subtypes, and we found the set A. So you can see the set A is, I think it's less correlated in the HER2 subtype, more correlated in the luminal B subtype, and the other variables don't show, in this case, right, with this ordering of the variables don't show a lot of difference. Okay, so it's the kind of thing we're after from the correlation matrix point of view. Okay. All right, so how are we going to do this? So if I'm given a data set and I want to go out and find a variable set A that's differentially correlated in the sense I defined before, how are we going to do it? I'm sure there's multiple ways of doing it. We chose an approach that we had applied in the past to community detection, and it seemed well adapted to this problem. So we do first of all, we start off with the obvious thing, right? We don't know R1 and R2, so we use the sample correlation matrices under the respective conditions. And then we essentially take that definition of delta IA I had before, and I stick hats over everything. Okay, so it's the sample version of the aggregate difference. So again, I'm looking at the sample correlation between I and J, the difference of the sample correlations in the two conditions, fixing I and summing over the variables in J. Again, I may be in J, the variable I may be in the set A or it may not be. Okay, so it's exactly the same thing, it's just the sample version of what I did before. So, our approach is testing based. So, and I'll explain how it works in a minute, but so the first thing we want to do is to ask, well, if I have a particular value of, an observed value of this difference, right, and I regard the quantity I've defined up there as the stochastic version, right, the random thing, then I can form a p-value in the usual way. So I can ask how likely is it under the null, and the null would be where there is no difference, delta IA is 0. How likely would it be under that assumption that the random variable delta hat would be greater than what I observe? Okay? So again, this is basically undergraduate stat. Is everyone okay with that? So that's just why this is useful, I'll mention in a moment, but that's the basic idea. So I'm going to look at this p-value. Now, in principle, you can't evaluate this in finite samples, even if you make a bunch of Gaussian assumptions, which we don't want to, it's still hard, and it would be really ugly. So what we do is the usual thing, again, classic, undergraduate statistics, is we say, well, all right, let's just find the central limit theorem. So with a little bit of work and not too much, and drawing largely on some classical results about CLTs for correlations and variances of correlations and correlation differences, right, we can approximate this p-value in this way. So essentially, right, when n1 and n2 are large and a is fixed, so I'm gonna highlight points in my talk where things are not so good. So here, as you'll see in the method, a is not going to be fixed. So this is the first weakness, if you'd like to point out. I think it's good for people to point out weakness. So weakness number 1, okay, a is fixed. Weakness number 2, clearly this is an asymptotic result, and if n1 and n2 are 15 and 20, how comfortable are we with this approximation? It seems to work okay, but, right, as I said, I don't have a theorem, and it would be hard to have a theorem that would hold in that case anyway. All right, but this is true. This is, this is a theorem. And there's a rather ugly formula for sigma hat, which involves, so it's important. We're assuming that we don't have differential correlation, but we do have correlation, right, under the first condition, and we have correlation under the second condition, and those empirical correlations end up getting plugged into here, right? So we do need to look carefully, right, at the structure of R1 hat and R2 hat when we're trying to compute this p-value, and that comes in there in particular. Okay, is everyone okay with that? So here's the method, and maybe I'll illustrate here. So let x1 be the data matrix under the first condition, x2 under the second, and I have some significance level alpha. So just point to this. This is the Benjamini-Hochberg step-up procedure, right? Or actually, I should say it's really the Benjamini-Hochberg. It's the modified Benjamini-Hochberg procedure when you can have under general correlation structure, right? So Benjamini, and I think it's due to Benjamini and Yekutieli. So they have a follow-up paper where they say, well, look, if your p-values are independent, which is the assumption made in the original 95 paper, then this works. If the p-values are regression, positively regression correlated, it still works. And in general, if you make this essentially a logarithmic correction here, then it works in pretty much in every case, okay? So we're doing something super conservative here. I just mentioned that. Now let me tell you what we're doing. So what we do is, again, this idea of a fixed point. So we start off with a seed set of variables. Now here's weakness number 2, right? This algorithm, which is, we love it, it's wonderful, it's a little delicate, right? If you throw it just an arbitrary set of variables, it kind of does the right thing. It's conservative, it returns the empty set, nothing, right? So in order to get it to return something, you have to start it at a nice place, okay? So we do a greedy swap. So we start off with a set of variables and we start moving variables in and out to try to get the correlation difference to be as large as we can. When we're done with that, we run this procedure, okay? So the second weakness of the procedure would be that you need to start it at a good place. But here's what it does. So I'm looking for a fixed point of the following setup, right? So and the fixed point goes as follows. So I compute these p-values, and again, they're going to be approximate p-values. I use the central limit theorem, right? I order them, right, the smallest. So this says that I1, right, is somehow the most differentially correlated with A sub t. I2 is the second most differentially correlated according to these p-values, all right? So if I wanted to collect together those things that are differentially correlated with A sub t, I would begin with I1, then add I2, and I would keep on going. At some point, I'd stop. Where do I stop, right? I just appeal to this Benjamini-Hochberg procedure, right, which is meant to control the false discovery rate at level alpha. So alpha is a free parameter for us. We typically take 0.05. All right, so I order them. This gives me a way of cutting that ordered set at some point k. There it is. I take that to be AT plus 1. Okay, so AT plus 1 is all the variables that were significantly, right, differentially correlated with respect to A sub t. right? And I keep on doing this. And so remember, at each point in time, I have a finite subset of, right, the integers from 1 up to m, right? So if you do this, then you know it's just, it's a simple mathematical fact that you will eventually get to a fixed point or a cycle, right? Because I'm just looking at, subset, subset, subset. If you keep on doing that in a deterministic fashion, and this is deterministic, right? There's no randomness here, right? Then you'll eventually get to a cycle or a fixed point. Typically, we get to a fixed point. The fixed point, right, that when you get to a fixed point, what do you know, right? So let's say that A is a fixed point, right? That means that everything in A is significantly differentially correlated with respect to A, right? Nothing outside A is significantly differentially correlated with respect to A, which is exactly the first part of the definition that I gave before, okay? So the fixed point of this search procedure, right, sort of puts us in the right place in regards to the kind of stable variable set that we wanted.
But does the set always decreasing?
No. No, so the sets A sub t, they can get bigger, they can get smaller, they can move around the space. There's no kind of monotonicity, which is as it should be. But yeah, is that yes?
I do not see why you need to order the variables. Have you tried to do it without ordering them? Would you get different?
You want to, we're trying to find, remember, ultimately, when we reach a fixed point, right, we want to be able to say that everything inside that variable set is differentially correlated with respect to the variable set. Everything outside the variable set is not, right? So that in particular, right, and again, what does significant mean? It means significant with respect to this FDR controlling step-up procedure. So that's going to be our definition of significant. So we don't want, so it's important that we look at the best variables. If we looked at a random subset, it wouldn't work. In fact, we would quickly end up, if that's what happens, if you choose a random subset to start this, it returns the empty set, all right? Because basically nothing makes the cutoff. So you really have to look, so it's greedy in that sense, okay? And ordering them means that when we get to a fixed point, we have something that at least satisfies part of the definition that we're after. Is that okay? Any other questions? So this is not, right? So we're using testing here, right? There's no estimation. I don't know if it's good or bad. Well, there's estimation in the sense that we've replaced R1 by R1 hat and R2 by R2 hat, right? But there is no other estimation going on here, right? So it's all based on this idea of a fixed point and iterative testing. So, and I'll show you, it seems to work well. And again, do I have a grand theorem that shows asymptotically that it's wonderful? Nope, right? We have some results that, under some assumptions that I don't really like, show that it should be conservative. That this thing that you get, the chance that you would find a non-empty set if you applied this to data where there was no differential correlation anywhere would be less than or equal to alpha. Okay, but I don't like the assumptions of that, so I won't claim that as being, a justification. But this is conservative. It will often produce nothing. And in my view, data analytically, I think it's better to produce nothing than to produce something that will lead someone on a wild and potentially expensive goose chase. My view. So, yes?
Unique?
Is what?
Is the set A unique?
Unique.
Yeah.
No. So in fact, we can find multiple sets A. And we do. So we typically find...
Also, I mean, empirical maybe, but also for the true set A is also not unique.
Right, it need not be unique. There could be several differentially correlated sets, and you can think about embedding them, especially if you don't let them overlap, you can think about embedding differentially correlated sets in a data set. So it's not, no, they're not unique. And typically we find 2, 3, 4, 5 in practice, rarely more than 5. Yes?
And then do you have a way to compare them?
We don't. So we don't, there's the first one that you find, but is it the best? So it's often the largest because it has the strongest signal. So which raises another question. Once you found the first one, how do you avoid finding it again? Right? Because, unless you could try to get your greedy swap procedure to avoid it, but what we do, and I'll say in a minute, we try to remove the signal. Once we found something, you could just say remove that set of variables altogether because look, it's a differentially correlated set, throw it out. But we want to allow that maybe these sets can overlap a little bit, right? And that seems biologically reasonable in some cases. So we don't want to do the hard thing of just taking the set, chopping it out of the data matrix, and setting it aside. So what we do essentially is to remove essentially the rank one, the principal rank one structure in the correlation matrices from X1 and X2, and then we rerun. And again, is it perfect? No. Little kludgy? Yes, so you can count that as weakness number 3. All right? How are we doing? Yes?
Is it somewhere built in now that the set you get cannot fall apart in 2 pieces?
Weakness,
That was one of the things you wanted.
weakness number 4, right? So what we do, no, that's, in fact, okay, I think this, I can come back to this, but that sort of takes me to the next slide. So first, maybe to get to the cycle. So you do see cycles, but rarely. If you see a cycle, I asked Kelly about this. I said, when do we see cycles? And so we've looked at a bunch of different data sets. You might see a cycle of size 1 is a fixed point, which is what we'd like. A cycle of size 2, you see occasionally. Cycles of size 3, rarely. If we see a cycle of size 3, we just stop. Okay? We throw up our hands and don't do anything. Maybe that's let's say, so we're now up to 4.5, right? Okay? If we see a cycle of size 2, then what Kelly does is she looks at the intersection, right? And they typically overlap, right? She'll look at the intersection of those two sets and she'll start the algorithm from there and then see what happens. But to address, that was okay, so that's handling cycles. I mean, that's, you know, it's a little bit ad hoc, seems to work okay. I won't claim that I have a theoretical, you know, that there's some great theoretical justification for that. Residualization, as I said, we essentially look at the correlation matrices separately in each data set under condition 1, condition 2, remove the rank 1 structure, and then from the data matrices, and then but again, only in the variables that we find. So if we find a differentially correlated set A, we only fiddle with the data matrices, right, and their values in regards to the variables in A. We don't touch anything else. And then we just repeat. Right. So the multiple DC sets is when we find something that's sort of kind of like 2 sets that have been stuck together. You think about sort of 2 diagonal blocks, right? Differential diagonal blocks in the difference of R1 and R2. In that case, what we do is we just run the algorithm on the restricted set of variables, and that usually reveals when we found these. But we do find these occasionally, and I think they can be biologically interesting. So we have a kind of, again, an ad hoc way of trying to pick apart stuff that's stuck together. Does that, maybe it's not the answer you wanted, but it's what we do. Is that all right? Okay. Any other questions? Yes.
So if you had in fact, the 2 correlation matrices, you directly would have subtracted and ordered them accordingly. But you cannot estimate the correlation.
Even if you looked at, if I knew the correlation matrices R1 and R2 and I subtracted them and ordered them, it wouldn't give me a DC set. Because there's a self-consistency, right? You remember, everything in the set has to be differentially correlated with the set, everything outside the set. And so if I just look at sort of a rank ordering the correlation differences, that won't give it to me. So it's and again, one can argue, why are you looking at this? It seems to us, obviously it serves us, the definition serves us well, and you can ask which came first, the algorithm or the definition, right? But the definition serves us well, and it seems, I think in practice, I have a sense that in practice it makes sense, right? It's because you're not just looking for a list of variables and you have to draw something. You're looking for a set of variables that are doing something together differently under 2 conditions. And again, without any scientific justification, it seems like that makes sense. So, is that a kind of a half answer? Okay. And then of course it's directional, right? So you remember, in the way we set it up, we're looking for things that are more correlated in condition 1 than condition 2. So when we've done all this, we swap conditions 1 and conditions 2 and just do it again, right? So we're looking for directional differences. That's also important. Okay? There's no absolute values here. And we don't want to, for reasons I can go into, we don't want to look for absolute values. It makes sense for us, again, scientifically, to look for just differences. High here, low here, and then we swap it around and look for, essentially high here and low here. Okay? All right. Okay. So, some features. So, we do not choose the size. The size of the differentially correlated set is essentially chosen by Benjamin-Hochberg, right? We do not say in advance how many sets we expect to find, how large we expect the sets to be, right? We don't say, well, we want to look for a set of 150 things. Well, there may be no set of 150 things, right? So we'll see in practice, it varies from thousands to tens, typically. We can find overlap because of the residualization procedure. I guess I can say it's fast, but I'll show you some timing results, and maybe you'll you'll disagree with me, but we don't need to aggressively filter the variables. We can deal with 100,000 variables and 1,000 samples, no problem. Okay, and we have software, but all right, that's not a big deal. Okay. So let me say a few words about competing methods, and I'll show you one of those pictures where we compare our methods to other methods, and lo and behold, we do pretty well, so that won't really be surprising. But maybe it's worth, this might be more important. So Kelly and I talked for a while about trying to find a way to sort of come up with a taxonomy of the existing methods, because there are plenty. So this is the best we could do. And this is not a complete list. So if you're the author of a method here and your method doesn't appear here, I apologize. These are just kind of representative methods in each approach. So one approach looks at identifying variables that have change in aggregate correlation across conditions, right? Which is a little bit like what you were suggesting. So you look at the variable in condition 1 and you look at its row of the correlation matrix and you compare, right? You compare that row with the same row under condition 2, right? And so that gives you a ranked list of variables, but it's not finding you a DC set exactly. Another thing was to look at clusters of communities under, say, cluster apply community detection or clusters in the first data set, and then see if any of those things look differently correlated in the second data set, right? But you're kind of doing something conditional there, and that's not, again, not an ideal way of identifying what's going on in general. And then there's the idea of just creating and analyzing, and should be, in this case, should be M by M. The M by M matrix capturing the differential correlation between conditions 1 and 2. It's the difference R1 minus R2, or R1 hat minus R2 hat. Notice, we do not, this is important, we do not compute that, right? I mean, I have it, so if you, this is a big deal because when M is big, if M is 100,000, which it is in some fMRI data, we look like you're hosed, right? You're hosed, or you have to go out and find a postdoc who really knows how to compute, right, and can figure out how to hold all the stuff in main memory, because it ain't going to stay in main memory, right, when you have a matrix that's that large, right? So the important thing is we make use of this difference. Let me go back here. But one more slide. So there it is, delta hat. So note that we definitely make use of the difference in those correlation matrices, but we don't need to compute them for every I and j, right? If we're considering the variable set A in our algorithm, right, then I need to look at every variable, so there's M of them, and compare its correlation with the correlations in the set J. The set J typically has small cardinality, much smaller than M, right? And I do this for a certain number of steps, typically 5 to 15 steps, and the algorithm will converge. So in the end, the number of comparisons that we do, the number of actual entries in that R1 hat minus R2 hat to be considered is really small, much less than M squared, right? It's of order M as opposed to order M squared, which is a big deal in terms of time, but also in terms of, space. Okay. All right. All right, so,
Andrew, in
yes.
approach 2, how are you defining clusters?
Oh, different ways. You can just look at correlation-based question. You can do it hierarchically. You can do k-means. There's a gazillion different things. I forget. WGCNA is, I think, they use it's hierarchical, agglomerative hierarchical based, and I forget what exactly what the splitting is done on, right? Because there's lots of, as you probably know, for clustering, there's lots of choices. So I don't claim that we've considered every possible clustering scheme. But the problem here is that you're looking at one data set, right? Doing some stuff, and then after, when you're done, post hoc, you kind of go and you say, I wonder if this is differentially correlated over here. So the best thing to do is to consider them both at the same time somehow. All right. So let me be brief here. So we essentially generate data. This is just simulating. So we simulate data in two ways. And the important thing here is in the first case, we make the data really nice. So the background is just, ID Gaussian, standard Gaussian, as nice as it could be. In the other case, we look at a single cancer subtype from the Cancer Genome Atlas, right? So we don't expect differential correlation. We split it at random into two groups. So here we have correlation. In the first case, we have very nice correlation matrices, empirical and obviously true correlation matrices. They're real nice. They're basically diagonal or close to diagonal, right? In the second case, who knows, right? We expect lots of complex correlations in real data, right? So that's our background. And then then we sort of induce, right, a DC clique or a DC set A by adding a fixed row vector to every variable in a set A. A is 100 variables. We let M be 1,000. Larger values of M actually knocked out some of the competing methods because they would just stop because they weren't fast enough. So, and then we looked at overlap. So to what extent does the empirical thing match the true thing? And then we said, well, look, it's easy to match the true thing if you produce a gazillion variables. So if your false discovery rate is more than 0.05, out you go, right? We set this to 0. So this is just a way of displaying what's going on. And then we did this for 100 simulations. And maybe, I guess the point I can say is, just don't ask me about this, I forget how that happened. But over here, what we see is that, in fact, WGCNA, the clustering-based method, it works pretty well. It beats us. Right? These are sort of correlation differences. So it's kind of signal strength. We've got signal strength along the x-axis and discovery modulo, not having too many false discoveries on the y-axis. And so we see that we do nicely in the case where we have a Gaussian background. Some of the methods are set to 0. That's because they're producing way too much stuff, right? Their false discovery rate is more than 0.05, so we set them to 0. But it's interesting, I think the takeaway here is when we look at a real background, right, which is what one expects to see in real data, right, so that the correlation matrices, even if they're the same, they're not simple, right? So R1 and R2 are maybe on the set A, they're the same, but they're not nice diagonal or banded or sparse, right? So then these methods kind of get a little wobbly, the other methods, and we do okay. So there you go. All right. Is that okay? Felt obliged to show a simulation study. I think more interesting, you're supposed to do this, right? Okay, done. That's all that. All right. Okay. This is more fun. So this is collaboration with some folks in the Cancer Center at UNC. They gave us some data. They were looking at antigen stimulation of B cell response. So if there's any immunologists out there, I hope you won't ask any questions. There's B cells and T cells. They were interested in B cell response. They were looking at late stage melanoma, and essentially they had a set of samples which they had followed up on and examined, and some of these samples had antigen stimulation of B cell response. I believe that was a good thing, right? That meant that the immune system was reacting to the cancer and trying to fight it, and that was the smaller set, 136, 325 samples. There was no antigen stimulation of B cell response. That was a bad thing. So there's 20,000 measurements, so we didn't filter at all. Maybe we threw out, there were a lot of zeros. This is count data, so I think we threw out maybe 5,000 genes or a few thousand, okay? And there's 461 tumors. So it's, it's a good sized data set, it's not giant. We found 3 sets, 3 differentially correlated sets, and you can see the numbers are moderate. There was no overlap, and the time, this is just in R, was less than a minute, okay? So I guess I call that fast. I'm an impatient person. That's enough time for, I go, I get coffee, I come back, it's done. I think that would be relatively fast. All right, so one reason that our colleagues were interested in this is that when they looked at differential expression, getting back to this point I said I would hammer on later, it's not informative. So apparently the differences, when they looked at differential expression, the differences in antigen stimulation in the, right, they're sort of responding and non-responding groups was confounded by a melanoma subtype. So breast cancer has subtypes, melanoma was the same. So they just didn't really see anything that was beyond what they learned from subtype. Okay. So here's what we found. So we found just the first gene set. So the correlation in condition 1 was 0.79, correlation in condition 2 was 0.65. So again, not like it was really high and 0, right? But we had, I don't know what you call this, high moderate versus moderate, something like that, right? And the gene set, they got excited. And again, I think it was the eyebrow. They passed the eyebrow test. They seemed happy. They did some gene set enrichment analysis, right? They said, well, all right, here's 138 genes. What biological processes do these have anything to do with? And they showed enrichment for, well, stimulation of B cells and T cells. To me, that seems like, well, isn't that what you expect? But I think the way they defined the groups, and I sort of oversimplified here. I guess I can use my pointer now. This isn't quite, when the way they measured antigen stimulation was by looking at certain, the diversity of certain sets of genetic clones. All right, so they had a set, and then they used an entropy measure to get at diversity, and then they looked at high entropy and low entropy groups. So that's actually where these came from. So these were not really based on gene expression, they were based on diversity of clones seen, I guess, in some follow-on experiment. So there was no, the response itself didn't have anything to do with expression. So I think that's why they were kind of excited, because what we came back with did have to, it made biological sense, and I think it was consistent with what they expected to see. Now, differential expression, nada. Right? So what we did here is we took all the genes, there's 20,000, a little more than 20,000, and we just ranked them according to their T statistics. And then we drew lines wherever we found something. These are 138 lines, and they're labeled. I'm sorry, you can't really see what's going on here, but each one of these things has, this is TRAF D1. All right? So, yeah, so some of them are differentially expressed. Some of them are not differentially expressed. You know, it's like the mama bear, the papa bear, and the baby bear. Some of them are in between, right? So there's no pattern, right? So that's good. It doesn't mean that what we're finding is, that alone doesn't mean that we're finding something interesting, but we're finding something different, right? So the point is the second order, maybe theme, you know, second order analysis reveals, right, which could be network-based analysis, is revealing something here that you can't find by just looking and saying which one of these genes, right, changes its expression between the two data types. So I think that's cool, right? And that, to me, this is a reason why we're all here doing network analysis, right? Because the models are more powerful, right? And they may not model everything, but they reveal stuff you can't find otherwise. All right. All right, one last application, and I'll go on to some other stuff. All right, the second example we looked at was fMRI data from the Human Connectome project. So we have about 100,000 brain locations, and we have voxels and gray ordinates. I think gray ordinates are on the surface of the brain. I Googled this today, and I got some fashion websites. So I clearly I'm way out of touch. Maybe is gray ordinate like a brand or something? I don't know, but go figure. So that wasn't very helpful. This is a single subject. They completed 316 language tasks, 284 motor tasks, and we found 5 sets of brain locations. Here, MATLAB, we used a MATLAB implementation, a bit faster, but again, 1 to 3 minutes. So, you know, we've got 100,000 by 600, and it's pretty fast. What do we see? A pattern. We were happy because we're not brain scientists, and I have yet to find someone who's been willing to look at the data and say that we found anything meaningful, but at least, unlike gene expression, right, here you can look at it, right? So we basically looked at the gray ordinates. This is, you know, your left hemisphere and your right hemisphere. And again, here, you know, moderate difference. We go from moderately correlated to more or less uncorrelated in this case. And so we plotted the 1,200 locations, and they have some spatial structure. And Kelly went and looked up some of this, and some of these places where the dots appeared are associated with language function and motor function. But again, I wouldn't put too much stock in that. But all right, so that was cool. And then we said, well, how about differential expression? And again, you see, no, that it's not as if there's no spatial pattern here, but it's different. So to make the point, yet again, right, the second order analysis is finding something different than the first order analysis. So this is first order, second order. Okay? They have some things in common, but they're definitely different. All right. Bonus material. All right. Okay. So I wanted to say a few words about a couple other projects, and really just a few. One is multi-layer networks, and particular community detection, which is something we've been interested in for a while. So for us, switching gears, a multi-layer network is just a sequence of graphs on a common vertex set. Vertices are registered, as has been discussed here. So the ELF graph has vertex set n, and edge set EL, which can vary. The vertex set n does not. The vertices are registered across layers, and the edge set, as you would expect, the edge set EL reflects, you know, different interactions between the vertices across layers. And again, a simplifying assumption here is we do not have edges between layers. So to some extent, if you permute the graphs G1 up to GM, it's the same multi-layer network. Okay, so we're not really looking at time evolution per se. Okay, so our goal here is really simple. So for us now, a community is not just a set of vertices, but it's a set of vertices and a set of layers, right? And we want to find a set BL. So right? Set of vertices, set of layers such that the vertices in B are highly interconnected in the layers L. Okay, so no surprise. All right, and just briefly what we do and what we can say. In this case, we, after thinking about it for a while, we decided the best way to approach this was through, a modularity-based score function. Okay, and so there's essentially the score function. This is modularity. We're taking the positive part because we're only really interested in sets B that have positive modularity. And so don't worry about the details, but essentially we have a result on score consistency. It turns out to be harder than one would expect to establish. Maybe we've done it the wrong way, but if you look at the empirical version of the score and you fix L, so again, this is simplified. We consider L being unknown, but if you fix L and you look at the empirical version of the score and you choose the set B that minimizes the score here, then you find that essentially that set Bn hat will have a good match, right, under a standard 2 stochastic block model with either community 1 or community 2. And we have...
What are DL of U? DL here.
Degree. So it's the degree of U in layer L. Sorry, I should have said that. Yeah, again, this is like hand-wavy stuff. But is that okay? So this is just the standard, nothing more than the standard modularity. Okay, so bonus material 1, bonus material 2. This is come up in versions of this have come up in a bunch of discussions that we've had, I know. So we're doing something, again, looking at a very simple version of preferential attachment. With apologies to those who know much more about this than I do, we're looking at a simple model, right? A generative process where we start with 2 connected vertices, we have an edge, and then we're going to add new vertices to this system, one at a time, and as it says there, we will attach our new vertex U to a vertex V at random, but in a way that is proportional to the degree of V plus some constant A, where A is greater than -1. So standard preferential attachment model. And as everyone knows, when we do this, we get a growing sequence of trees in this simple model, and we were interested in considering this from the point of view of a change point. And the only parameter here is A. So what if we run this process with a parameter alpha for a certain amount of time, and then after a certain time we switch and we run it with a different parameter beta. So that's the very simple problem that we were interested in. So here n is going to be, the number of times that we run the process is large, gamma is fixed, and it won't vary with n, some number in 0, 1, and alpha and beta are the respective parameters. Now, in our case, we get to observe the evolution. So we're not just given, I know this is another thing people have discussed, we don't just get to see the last graph, the last tree, we get to see the entire sequence, and we're interested in estimating gamma. So, 2 basic results. One is that the tail degree, the exponent of the tail degree distribution. First of all, the tail degree distribution exists. It's obviously, it's well known that it exists in the case where we don't have a change point, and not surprising perhaps that it also exists in the case where we do have a change point. What is a little surprising is that the degree exponent doesn't feel the change point, so it depends only on alpha. Well, let me say a few words about that. So again, we have the change point model that I described before. Let n of k be the number of vertices of degree k in tree n. So again, it's a classical result. It says that n sub n of k divided by n converges as n tends to infinity to some probability mass function. The same thing happens here, right? So we have convergence in probability to some PMF P of k. And importantly, P of k, we have bounds on P of k that depend on alpha, not on beta. So it doesn't matter if beta is bigger than alpha or less than alpha, regardless, right? And of course, remember, gamma is greater than 0 and fixed. This is important. If that weren't true, then this would not be true, right? But if we have a fixed change point, then essentially we don't see the degree distribution depends principally on alpha. These constants will depend on beta, okay, and gamma, okay? But the dependence is the same as you would expect if you just had run the process with parameter alpha the whole time, all right? And lastly, We do, and this is again very hand wavy, but we do have a change point estimator, and I won't tell you exactly what it is, but the intuition is this. If you just look at the generative model and you say, look, if alpha is less than beta, or vice versa, we expect to see fewer leaves after the change point. So somehow looking at the fraction of leaves, right, at a particular time and going forwards or backwards from that time, right, we would expect that would tell us something about where the change point is. So essentially what our estimator is this. So gamma n hat, again, more or less, is that gamma in 0, 1, such that the difference between the average proportion of leaves before time gamma n and the average proportion of leaves after time gamma n is maximized. Right? And this requires that we sort of understand the variability of those averages, right? So this requires us to use a Martingale functional central limit theorem, something else that I know people have mentioned here. So it's more technical than I think we imagined it would be at the outset, and essentially what we can show, again, with an estimator that's defined not exactly that simply, we can show that we get a rate of convergence like this. Yes?
So Andrew, what's the intuition that the rate of convergence of your change point is so much slower than the usual parametric 1 over n?
That's a good question. The usual, well, all right, so we're not in a standard parametric setting here. We're not seeing I, so I think part of it comes from the dependence and the complexity of the model. I mean, that's kind of that's a cheap answer, but that's my best answer, and it just requires that the model doesn't allow us to carry out. If you look at the kind of stuff that Ed Carlstein had done years ago, right? And then let's see, Lutz, Dumgin, right, and other folks. So they had gotten very tight rates. Those arguments just don't go through in this case, and you're forced to appeal to fancier machinery and central limit theorems. So it's maybe not unsurprisingly you get a root n rather than an n there.
So my next question, is this the optimal rate or it's a limitation of the technique?
That is a good question. I don't think, we don't have any results on optimality. So it might be that you could do better. I'd be surprised if you can get rid of the root n, but maybe you can. But I don't. I suspect that one might be able to do better than to reduce the logarithm. Maybe get rid of it or replaced by something that's growing more slowly. So
Actually, I'm not surprised by the log n. I see it in stuff that I'm going to talk tomorrow.
yeah. Okay. So
Then I'm not surprised.
okay. So just a summary of what I talked about. Testing-based approach to differential correlation. I hope that was interesting, if not theoretical. So again, I guess the takeaway message there is that there's, I don't think I have to tell anyone in the room that looking at second-order different changes of some sort, right, provides different information, sometimes more information than you can find by looking at first-order effects, and apparently it's in some cases at least of scientific interest. The procedure that we use to identify these DC sets is a little unusual. It's testing based. I guess it's iterative testing based. It doesn't involve estimation. I guess that's worthy of mention. Talked about some applications and just the bonus material I just described. We're able to. I think we have a decent method for finding communities and multi-layer networks. Again, without connections between the layers, and perhaps these are worthy of note. Thank you very much.
Are there any additional questions for Andrew? I have
Yeah.
for the first part. I mean, you have a parameter alpha.
Yes.
This is parameter alpha an interpretation, some other coverage probability.
Yeah. So, all right. So, where to go? Right. So first question is, the first thing I would say is that the performance of the algorithm, it's Lipschitz and alpha. I mean, Lipschitz with quotes, right? It's not like you change alpha from 0.05 to 0.06 and suddenly, you get crazy new results. So the results don't change. If you turn up alpha, you're going to get, if alpha's larger, right, then essentially you're being less stringent here, right, and you start to see typically larger sets. So the choice of 0.05 is just, I don't know, sort of a nod to Fisher, I guess. I can't say that it's more motivated than that. We've tried different values, and it doesn't seem to make a big difference if you're in a range of 0.05. So here, I think we do not at this point, have any result that says, look, the chance of finding a set, if I have data, as I said before, we have some results under conditions I don't like that say, if you have data where there is no DC clique and you find one, the probability of finding one with this procedure will be at most alpha, right? So there's some results of that sort that we've talked about, but they're not concrete enough yet for me to put them on a slide. So I think alpha might help us try to quantify how, assign some kind of a p-value to the set that we find at the end. But I think we're far from having a general result that says that. So really, I would think of this, the use of p-values here is methodological. I spoke about this in DC, and people, someone in the audience didn't like p-values. Surprise, right? So they said, oh, this is that anything that with a p-value must be bad. Our use of p-values here is not so much to draw an inference or tell someone to accept a hypothesis or reject a hypothesis. It is purely methodological, and to that extent, the parameter alpha here is just helping us. It sort of helps us tweak our methodology, and so, but we typically send it to 0.05 and let it run. I'm not sure if that's so I don't think it can be assigned. I'd like to be able to say more about it than that, but I don't know if it has a deeper meaning than that.
Any additional questions?
Yeah.
Well, also a question about the same. So your criterion was about the R's, the correlations.
Yes.
And then you make the step to the p-values.
Yep.
And is that warranted because all the sample sizes are the same, so that the ordering, the presumed ordering between the R's will be the same as the presumed ordering between the P's? I mean,
we don't remember. So it's important. The p-values here involve a point, a variable, and a set of variables. So we're not ranking. So importantly, we're only ranking variables within the algorithm for a fixed set relative to a set A, right? So it's not like we're never looking at Pij. We're looking at Pia, right? P1a, P2a. So it's a little bit different. So we're not trying to capture, in that sense, we're not trying to capture what's going on with the true correlations. It's always relative to a set A.
Then can you comment on the difference? So now you've got the sum of correlations.
Yes.
Could you think of a criterion where all correlations should be positive, or all differences should be positive?
Right.
And what would be the difference between aiming for a sum that is positive or aiming for everything being positive?
Well, if we're in the situation where really all the correlations, all the true correlations are large and positive in data set 1 and basically 0 or negative. Well, they can't all be negative, but basically 0 in data set 2, then the procedure could find a giant, it would find everything or it would tend to, right? And happily that doesn't happen. That's not really useful. Of course, most data sets don't have that feature. I'm not sure if that's so right.
So I was wondering about the relevance, so the biological relevance of the sum of the correlations. So why is the sum of correlations something relevant?
I will not claim that I would not assign a biological interpretation to the sum per se. I think it's a way of finding things that are here's my biological story, and this is just definitely a story. So we imagine that there's some network, or people use the term network and pathway. Right. So we have a network or pathway that is operational in healthy people, right? So the genes are correlated to some extent, right? They have a coordinated activity. It's doing something important for the cell. Right? Maybe it's cell death or cell growth or energy, whatever, right? That same network has been disrupted in the sick patients, and those genes are now their activity is no longer coordinated. They're just going on their merry way. So it's in that rough sense that we feel like looking at the sum of correlations is good. You could look at absolute values. You could look at other measures of association. Again, our feeling was my philosophy is dumb first, and then then you can do the clever stuff second. So looking at the correlations seemed to be the best thing. But I'm not saying you can't do other stuff. Yeah.
Okay, so let's thank Sandy again.